Eighth module on the series of Bioinformatics for Scholars 2023. The title for this module is Alignment and Trees. The module would introduce you to the concept of sequence alignment, which would include pairwise sequence alignment, multiple sequence alignment, and also understanding the conservation of sequences and how multiple sequence alignment can be used to understand the close and distant relatives. This module would also introduce the concept of phylogenetic trees and its usage. The first session on alignment and trees module is pairwise sequence alignment. This session will include slides to brush up the concepts of what sequences are, why sequence alignment is needed, what is sequence alignment and how we go about it, what is pairwise sequence alignment, and what are the strategies used in pairwise sequence alignment, types of alignment and algorithms, a demo on the tool to run pairwise sequence alignment, which in here we would be using a tool called BLAST, which is most widely used tool. So as you have already learned in the previous session, we know what are sequences, right? This slide is just a recap. A genome is an organism's complete set of genetic instructions. Each genome contains all the information needed to build that organism and allow it to grow and develop. Central dogma is the process by which the instruction in the DNA are converted to RNA, and then it is converted into a functional product, usually a protein. This involves DNA instructions being converted to RNA message, which is then translated to produce a protein called protein, which contains a strings of amino acids. What is DNA, RNA, and protein? Let's dig a little deeper into this. Your genome is made up of a chemical called DNA or deoxyribonucleic acid. DNA contains four basic building blocks or bases called adenine, A, guanine, G, cytosine, C, and thymine, T. The order or sequences of these bases determines the information available for building and maintaining an organism. Similar to the way the letters letters of the alphabets appear in a certain order to form the words and sentences which gives a meaning. DNA is a two-stranded molecule which has a unique double helix shaped like a twisted ladder. DNA is transcribed into a molecule called RNA or ribonucleic acid. RNA plays a key role in turning the instructions held in the DNA of your genome into a functional protein in your cell. RNA is closely related to DNA, but it contains a different sugar called ribose and the base uracil replaces the base thymine in DNA. The other bases are common in both RNA and DNA. The RNA structure is completely different, which is a single helix. RNA is then translated into a large complex molecules called proteins. These large molecules are made up of chain of amino acids. Proteins are essential to the functioning of our cells and play many critical roles in the body. There are 20 different amino acids which are the building blocks of proteins. Similar to DNA sequences, the protein sequences with these amino acids determines the information available for building and maintaining an organism. As I said, DNA, RNA, and a protein all have a stretch or a sequence of their building blocks, either bases or amino acids. Based on the arrangement of these bases or amino acids, much information can be inferred. One such way to understand these sequences is sequence alignment. Alignment is a process of comparing a known stretch of DNA or protein with another to check how similar these two sequences are. Based on the similarity level, common relationship can be inferred. Here is a short stretch of example of a DNA align. As you can see on the screen, during this process, a number of gaps are introduced in this alignment. Why? Because we know that no two sequences can be 100% identical. If they are, they can't have two different functions. But to find the highest similarity possible, these gaps are introduced between the sequences and found the highest possible similar sequence with the query sequences. Okay, now we know what a sequence alignment is. But why do we do that? There are a number of reasons and a number of information that can be gathered from a sequence alignment. First, we can identify the regions of similarity between two sequences. From the image given on the slide, if you can consider each block as a small stretch of sequence, the stretches that match can be spotted. In the case of protein sequences, the stretches could be an alpha helix or a beta strand structure. If you align a parent and child's DNA structure, these stretches could be identical sequences passed on to the next generation. Next is to identify the sequence similarity. Given two sequences, the percentage of similarity between these two sequences can be identified. As I said in the previous slide, sequence alignment can be used to identify the stretches that are being transformed to the next generation. When aligning more than two sequences based on the similarity in these sequences, a phylogenetic tree can be constructed, which is a way to visually represent the evolutionary relationship between sequences, about which we would be learning in detail in the next session. Another important application of sequence alignment is developing protein models. This is applied when a new protein sequence is identified. When a protein function is unknown but the sequence is there, we can align the sequence with all the known protein sequences in the family and identify the structure of protein from the most identical sequence. These information we gather from sequence alignment can turn into various applications such as identifying a function or a structure of the newly discovered protein or gene. Based on the identical regions, a set of functional patterns and domains can be identified. A great example for this would be our genomes. A genome has only 2% of exons, which is later transcribed into RNA and then translated into protein. The rest, 98% of the genome, is introns or junk regions, which protects the exons, which just protects the exons. Based on this sequence alignment, functional patterns or domains like exons can be identified. The most important application of sequence alignment is to find the evolutionary relationship between individuals or organisms. Now we know what sequence alignment is and its importance in any biological study. But how we do that? There are two types of sequence alignment. First one is pairwise sequence alignment where we align only two sequences. One would be the query sequence and other would be the target sequence. In this case, the query sequence is aligned against a known target sequence. The second type is multiple sequence alignment, where more than two sequences are aligned. We would be learning multiple sequence alignment in detail in the next lecture. Pairwise sequence alignment. As I said, it is a process of aligning two sequences. From the image given here, you can see the sequence 2, which is a query sequence, is aligned against the sequence 1, which is a target sequence. Based on the relatedness of these two sequences, information on query sequence can be inferred. As you can see from the image, Gaps are introduced on the query sequence to increase the concordance between the residues. Here, first nucleotide A matches in both the sequences. Second, there is a mismatch, and third and fourth are matched. Whereas in fourth case, there is a gap introduced so that the sequence alignment can be better and gives a better scoring scores for these sequences. To find the best pairing of two sequences, two different strategies are followed. These are called global and local alignment. Global alignment is done for the entire two sequence, whereas local alignment is done for specific stretches on both the sequence. Uh, Global alignment is an end-to-end alignment strategy. It aligns each letter in each sequence once. This can be applied for sequences. This cannot be applied for sequences which are divergent. Human to human can be aligned with this method, but with other organism it cannot be done. And it also not align sequences of different lengths. This is, as I said, this is applicable to align two closely related sequences of same length, not applicable for divergent sequence and sequence of different lengths. Whereas local alignment is a method to align shorter regions within two sequences. This method is used to align more divergent sequences. The main goal of this sequence alignment methodology is to find the conserved regions of DNA or protein that is present in two sequences that are transferred to the next generation or the offsprings. Okay. The images that we were showing till now had not more than 10 bases in a sequence, and hence we were able to just look and say how much the sequences were similar and how much were not. But in real time, that will not be the case. The sequence length will be much bigger. In that case, we would be using tools to check the similarity between sequences or align the sequences. To align two larger sequences, a set of steps would be followed by tools called algorithms. What is an algorithm? To instruct a computer to do something, you need to specify the steps to it. Like you have a recipe to make some food. To make your food, you first need the ingredients and then you give a proper instruction of how it has to be made. The same way, for a computer to run a program, you should provide the instructions line by line, and this is called as an algorithm. Global and local alignment are fundamentally similar, and hence the steps followed are more similar in tools. There are three different algorithms or steps followed to do sequence alignment. First is the dot matrix method, which is the basic strategy to define a computer to do sequence alignment. Second is the dynamic programming method, which is the most frequently used method for its accuracy. Third is the word method, which is mainly used when one query sequence has to be aligned to a number of sequences to find the best match. Digging deeper into the first method, dot matrix method is a basic method for all other sequences, for all other sequence alignment methods. In this method, both the query and target sequence are placed horizontally and vertically in a matrix. A dot is marked on all the boxes where the horizontal and vertical bases are matched. Finally, a diagonal line is drawn from the right bottom corner to the left top corner. The line breaking the points are considered as gaps in the alignment. Once these lines are completed from right bottom corner to top left corner, the basis from starting of two sequences is wrote down to see the gaps in alignment, and then the alignment is actually given as we saw on the previous slides. The next is the dynamic programming method. This method is similar to the dot matrix method, but instead of dots, a score is given for each matching basis. As you can see in this method also, there is a matrix where you keep both the sequence on a horizontal and vertical way, and then wherever the scores are matching, wherever the bases are matching, a score is given. This method provides more than one sequence alignment possibilities based the highest scored ones are considered as the best at the end. The third method is word method, which is completely different from previous methods. The method is developed based on the assumption that any two related sequences might have at least one word in common. In this method, first, short stretches of identical letters in two sequences are found, and the rest of the sequence are placed for alignment based on the short stretches. So here you can see a target sequence is placed and the query sequence is placed, and after that all the possible short stretches, by keeping it a little ahead of the sequence, a target sequence, all the possible short stretches are found, and then finally the alignment is alignment is placed based on all the short stretches that we have found. So here is a here is a tool that we will be using for demo here to see how pairwise sequence alignment is done. Basic local alignment search tool is uh, is is called as BLAST. BLAST is most used tool for sequence alignment. BLAST finds regions of similarity between biological sequences to the sequence database. Here we will be using this BLAST tool to do a pairwise sequence alignment. As I said, BLAST is a heuristic algorithm for performing database searches. There are different programs available in BLAST tool. One is BLAST-N, where a nucleotide sequence is aligned to another nucleotide sequence. The second one is BLAST-P, whereas a protein sequence is aligned to another protein sequence. There is another method also called BLAST-X, where when you give a input nucleotide sequence, it is translated into protein sequence, and then this protein sequence is aligned to all the possible protein sequences. So here is the URL given for a BLAST tool. I'll also be showing you how to do this. So to start that, so here is the BLAST tool that I have opened. You can also type BLAST on Google, and the first link that you get would be the basic local alignment search tool, that is BLAST. And here we are going to use, we will be using protein blast. So here, as I said, there are different types of blast algorithms available, specifically for nucleotide alignment, protein alignment, and translated nucleotide to protein alignment. So here we are using protein blast for this demo session. So when you click into protein blast, you can see there is a space to enter your query sequence, and then there are other informations given. So generally, BLAST is used to align one query sequence against the database sequence. But here, as we are going to use do primary sequence, I mean pairwise sequence alignment, we have to click on align two or more sequences. When you do that, you will get an option to to paste two different sequences. So this is either you can give the accession number or the FASTA sequences. You know what is FASTA sequences. You might have learned that from the previous sessions. Here I have two FASTA sequences. One is one is the hemoglobin subunit alpha of human, and the second one is hemoglobin subunit alpha of mouse. So I'm using these two sequences and I am aligning these two sequences. Here, make sure that all the FASTA format is proper in the sequence that you have given. So it should be the first line should, as you know, FASTA sequence, first line should be the head, like title or the header, which starts with the greater than symbol, and then is your sequence. So here, after pasting these two sequences, I'm just clicking on blast. Since the sequences are very small, we would be getting the results sooner. Here is the graphic summary of alignment. So this is what we want to see here. So here you get all the information of about the sequences, its scores, gaps, and everything. So if you see the query 1 is blasted against query 2, wherever it has matched, you can get in between the nuclear, I mean amino acid is written in between if it has a match, and if it does not have a match, there is a gap. So wherever so totally they have introduced two gaps. We had 143 nucleotides in one sequence, and I mean amino acids in one sequence, and 141 amino acids in other sequence. So the identity between these sequences are found to be 72 out of 143 amino acids have been identical. Hence, it is 50% similar. These two sequences that we gave is 52% similar, 50% similar, and there have been two gaps introduced, and the rest everything is like these are the informations that you see from a pairwise sequence alignment, and you use these informations for your study. Mm. Mm. And you can see a graphic summary here where query 1 is placed against query 2, and here it's like 80 to 200 nucleotides have been aligned. And if you want a detailed description of how much it has, here is the maximum score, total score, and query coverage 100%, and the percentage of identity is 50.35%. All these informations are given here in this thing. So this is the basic information on subject length and so on. The rest information as I showed, but this alignment is the most important thing, where you just see how these two sequences have been aligned in your query and subject sequences, and the identity and how much positive and how much gaps have been introduced in these two sequences. These are the informations that you see. Points to remember, which is a pairwise sequence alignment is a fundamental concept for many bioinformatics applications, which provides inference for the relatedness of two sequences. Strongly similar sequences are often homologous. Sequence alignment strategies include local and global alignment, and there are three types of algorithms, which is dot matrix, dynamic programming method, and word method. Dynamic programming is the exhaustive and quantitative method that is mostly used method. In this session, we would be looking at the overview of what is multiple sequence alignment, what are the applications of multiple sequence alignment, steps involved in multiple sequence alignment, and how to build a phylogenetic tree using multiple sequence alignment, and what is evolution, what are the forms of phylogenetic tree representations, and how do we construct a phylogenetic trees, what is the steps involved in that, and then there is a demo of a tool called Clustal Omega, through which you can do multiple sequence alignment and also build a phylogenetic tree. So, multiple sequence, as we all saw in the previous slides, multiple sequence alignment is a method to align more than two biological sequences. When you have a subject sequence and two more query sequences, multiple sequence alignment method is used to find the similarity between all three sequences or all the four sequences or how many ever you give. The image given here is a pictorial representation of multiple sequence alignment. Here are three sequences. Consider these as three sequences. Each box in each of these lines in a different colors are bases of nucleotides maybe, and you find all these sequences are aligned to find the highest similarity between all the three sequences. So, like pairwise sequence alignment, here also we can introduce gaps to get a better alignment between the sequences. So there are a number of applications for multiple sequence alignment, and most of them are similar to pairwise sequence alignments applications. First is the detection of structural homologies. When you have a new protein sequence for which even the family of the protein or any other information is not known, the structure can be predicted by running a multiple sequence alignment between all the sequences available on database and your query sequences. And this multiple sequence alignment can also help in improved prediction of the structure of the proteins. As we all know, the important insights of a protein or DNA sequence lies on the conserved regions or motifs of their sequences. Through multiple sequence alignment, we can find these conserved regions that are similar in sequences. The basic application of multiple sequences is as usual to find the similarities or detect the similarities between sequences. Based on the conserved regions found, we can build evolutionary trees or linkages of these sequences to understand how similar are these sequences and what is the ancestral sequence of all the sequences available. So there are basic small steps behind building a multiple sequence alignment. So we can't directly just take all the sequences, put it on a row and align them, because it's not feasible. So here what we will do is, how many ever sequences you get, we try to compare all the sequences pairwise. For example, if three input sequences are given, sequence A, B, and C. Sequence A is first paired with sequence B and done, and a pairwise sequence alignment is constructed, and then sequence A is again paired with sequence C, and then A to C pairwise alignment is done, and then B and C pairwise sequence alignment is done. So we try to find all the possible combinations of pairwise sequence alignment, and out of all the possible combinations, we perform a cluster analysis on pairwise data to generate the hierarchy of this alignment, and then we build the MSA by first aligning the most similar pair of sequences, and then the next most similar pair, and then it goes on. So here is a basic example of how we do a multiple sequence alignment. Here are three sequences: sequence 1, sequence 2, and sequence 3. First, you build a pairwise sequence alignment for all the possible combinations. And here is an example of building a pairwise alignment between sequence 2 and sequence 3. And then you try to build all the possible clusters that can be combined and found through these sequences. Once that is done, we take the most highest scored pairwise sequence alignment. In this case, we have got sequence 2 and sequence 3. We put them on the horizontal row of the matrix, and then we put the other sequence, the next sequence, on the vertical side of the matrix, and then again we build a build the matrix for the pairwise sequence alignment. Once that is done, we align all these three sequences to find the final multiple sequence alignment for these sequences. So, once multiple sequence alignment is done, we build a phylogenetic tree, which would be a representation of the multiple sequence alignment. So to see how similar are these sequences. In the above slide that I gave, there are only three sequences. We were able to just look at it and say that this is similar, this is similar, this is similar. But in all if you have number of sequences, for example, here there are like 5 sequences in this ancestry. You can't just go and check each of the nucleotide and say how similar are these sequences. So in that case, we built a phylogenetic tree as a representative of multiple sequence alignment to find the most similar sequences and then how these alignment has been built. So what is the advantage of building a phylogenetic tree is to study the evolution. So, for example, if you have 3 or 4 organisms, you want to see how similar are these organisms, you go for a phylogenetic tree constructions. So, what is an evolution? Evolution, in simple terms, is the change in the organism over generations. So, through natural selections, it origins to a current existing forms, and unfit forms are eliminated via environmental changes. The underlying mechanism behind this evolution is genetic changes and genetic mutations or changes. That is why we try to find the most similar sequences through these genetic sequences or protein sequences. So, phylogenetics is a study of evolutionary history of living organisms, and phylogenetic tree is a diagrammatic representation of evolutionary relationship among organisms. The patterns of branching reflects how species evolved a series of common ancestors. So here, the nodes on each of these clusters or trees is considered as the common ancestor, and the branches represents the length of the deviation between these sequences. So there are two forms of representing a phylogenetic tree. First is cladogram and second one is phylogram. A cladogram is the way of just lining up the external taxa, and this branch lengths are not proportional to the deviation between the sequences. It's just a representation saying that these sequences are similar and these are not. But phylogram represents the length, the amount of evolutionary divergence between the sequences, and information about the relevant divergence time and times are represented through branches. If you see the images given here, you can see all the cladogram. In the cladogram images, all the endpoints are straight, whereas in phylogram images the endpoints are not straight, representing that how much deviant or how much deviant it is from the sequence that is aligned together. There are certain steps in constructing a phylogenetic tree or algorithm that we follow to construct a phylogenetic tree. First thing first, we have to identify and acquire a set of homologous DNA or protein sequences. There is no point in running a my multiple sequence alignment and then constructing a phylogenetic tree for a sequence which is completely deviant, because there you won't get any similarity between them. If you run if you run on homologous DNA or protein sequences, you can find the smallest of differences on these sequence between these sequences. First, we perform a multiple sequence alignment on this on the query sequences that is given, and then we determine which method of tree building you need to do, either cladogram or phylogram and so on. And then you estimate a tree from the aligned sequence, and then present the tree in a way to convey the relevant information. So here is the tool called Clustol Omega, which is widely used method in molecular biology, which works on progressive alignment method, and most closely related sequences are aligned first, and then the group of sequences are added as mentioned in the algorithm of multiple sequence alignment. This Clustol Omega provides a phylogenetic tree for the multiple sequence alignment. It performs both multiple sequence alignment, and also it provides a phylogenetic tree in the tool itself. So here we are going to see a demo of how to run a multiple sequence alignment and then construct a phylogenetic tree using the Clustol Omega tool. For that, in Google, if you just search for Clustol Omega, you will get this first Clustol Omega tool, which is run by EMBL EBI. And when you click on this, this is the Clustol Omega page first that opens. Here we are going to run multiple sequence alignment for the protein sequences. Since here is already protein given, if you want to run for DNA sequence or if you want to run for RNA sequence, on this tab you can change, and then you can give your query sequences. In our example, we are going to run it on the protein sequences. So I'm just keeping it to protein, and then there are two options here. Either you can just paste the sequences in FASTA format, or you can choose to choose a file that in which you have saved your sequences. In our case, I'll be pasting 4 sequences of 4 sequences, and so here are 4 FASTA sequences. So this is alpha globin subunit structures sequences, and then directly you just click on submit. Here is step 2, your parameters. So Clustol W with character counts, or Clustol W, we are going with Clustol W with character counts, and then we are submitting it. So you will get a page like this. Your job is queued and will be running shortly. So it will take a few minutes to run multiple sequence alignment and then give you a phylogenetic tree. Here I have already ran this and kept so that I can show the results. So this would be your result page that you get from Clustol Omega once you paste your structure and run alignment. So the result page will have a number of tabs like alignments, result summary, guide tree, phylogenetic tree, result US, submission details, and the download option. There is an option to download your alignment file as well, and there you will get an option to download your guide tree as well. So first, if you see this alignment, you can see we have aligned 4 sequences, and all the 4 sequences are pasted here, and the length of sequences are given. First sequence is 143 amino acids, second, third, and fourth as 142 amino acids. So this star represents that all the 4 amino acids are same, and it is aligned. And then this double colon represents that 3 maximum of them are aligned and few are not aligned, and the gaps are places where it is not aligned, and similarly dots is also where few differences have been found. So this is your sequence alignment, multiple sequence alignment. When you see 4 sequences, since this is a shorter sequences that I've given, we have We can see this in short way. When the larger sequence is given, it will be running on. So next is result summary. So here the information on how many input sequences have been given, what sort of guide trees and everything is built. So now to guide tree. So as I said, there is this cladogram where all the endpoints are equal and it does not represent the length of it does not represent the branch length as the deviation from the sequences. Whereas if you click real, you would get the tree where the length represents the distance between these two sequences. This is how a phylogenetic tree would look like, and this is your first sequences, sequence 2, sequence 3, and sequence 4. And then okay, you can also see the result submission details, that number of sequences submitted and the input and output sequences and so on. Result summary gives you all these details. And the most important thing to check in a cluster omega is alignments, and the guide tree or phylogenetic tree, both are same. You can just see that, and if you want to see download your aligned file, you can download it from here. There is an option to represent this in colored format also. Each of this red would represent how much they are similar and deviant. So these are the options, and this is how multiple sequence alignment is done, and phylogenetic tree is constructed. So points to remember from this session is the best approach is to perform alignment using a combination of multiple alignment programs. Molecular phylogeny is the basic tool to understand sequence evolution in phylogeny. It is important to understand the assumptions and limitations while creating an efficient phylogenetic tree.